lecture on study designs. So study designs. Mr. Abdullahi, what is study design? Yes, please. Asamaba. Okay, Castro. Mr. Castro. Is he around? But we have 9 people here. So, okay, let me call them. Fast, because the time too, so let's do it fast so that you can go to wherever you are going. Abugre Abdullah, what is study design? Isaka, study design. Hello, Mr. Abdullah, it seems your people are not around though. What is study design? What is study design? What is a study design? You people have to have idea. Alfred Aji, study design. Alfred, you put it anywhere. Just put it anywhere. Yes. eh. Oh, okay, that's good. That's good. Inusa, your hand was up. What about David Brigrini? David. What do you have for me? Okay. That's why I'm asking him, and he said there's no idea for now. But he raised his hand. Hey, so questionnaire. So it's right, Okay, Immaculate to give us the right answer. Immaculate. Answer it and let's start the class. Immaculate, answer. What is a study design? Immaculate, hurry up. Okay, so let's go and start the study. Let's go and start the lecture. Okay, so please mute your mics, Can you all hear me? So then mute your mics if you have a question, because this is very technical. It's research methods. I would have recorded for you, but you need to also put your inputs so that you understand from the lecture. Then when you get if you get the recording, fine. But if your reaction is not good at the end, I will not send you any recording. That's how I do my things. Okay, so study. If your reaction is not good and you people are doing hide and seek, I will not send the recording. Well, you are here. Okay, so epidemiology. Please mute your mics, eh? I'm starting. I'm recording. You shouldn't be talking on the background like that, please. Okay. So, when we say epidemiology, it's just used to define the dynamics of a disease. So it's, and public health is a subset of what some people will say epidemiology is a subset of public health. Some also say public health is a subset of epidemiology. Whichever way, we should confuse each other. Okay, so it looks at the distribution or the dynamics of diseases. It also looks at the causes of disease, and also when you do a study, it looks at study designs, causation, that's causal relationships, and those things. We will be doing all those things. It looks at bias and other things. So, for public health, you look at biostatistics aspect and also the epidemiological aspect. So, study designs. Before you start a study, what do you do? How do you design the study? How are you going to recruit your subjects? Are they going to be longitudinal? Is it going to be cross-sectional? Is it only one person, like you are 1983, when HIV, when they were trying to diagnose HIV anew? What happened is that, you know, when a doctor finds a condition which is so abnormal, so rare, the condition is very rare, like it doesn't often occur. Then the doctor will write the findings and find out whether the patient will die or will survive. Then he will write a paper on it or a report on it, and that is what we call case report. So we have different types of study designs. That is how you group your study, how you design your subjects. Whether you are going to work in pregnant women, then which condition are you using? Is it the pregnant women who have malaria and those who do not have malaria? You're going to compare them, and that one becomes what, either a cross-sectional or a case control. So let's go through the step by step. So we have evidence of hierarchy of epidemiological study designs. So when somebody says that he has gotten or he's doing a research, the research is in hierarchy. Weight of evidence means that like every research you are doing or you conduct tries to prove an evidence as to whether what people have found is different from what you have found. For instance, you want to prove evidence that a drug that you have discovered is able to cure coronavirus. So that is your weight of evidence. So you have to choose among all these, any of these study designs. So the study designs are case control reports, case series, ecological studies, cross-sectional studies, case control study, retrospective cohort studies, prospective studies. Prospective is the same as longitudinal study. Then we also have randomized clinical studies or randomized control trials. Then we also have the unrandomized control trials. So we'll be looking at them step by step. So in any study, the base of the triangle, here shows the weight. So the research, the base of the triangle shows the weight. Class rep, please tell the person. I can't see them. I can't see the person who is talking back at the background. Please try and contact your people to stop that. I want to see let me see the person. Sorry for everybody's muted now. Okay, so let's continue. So at the base, this is the apex, that's the highest, so highest evidence. So that's why we are showing with arrow. And this, from the case report to ecological studies, We call these three studies descriptive epidemiology or descriptive studies. They just describe, they don't infer any causation or causal relationship. When you say causal relationship, that is cause of a disease. Does snake bite lead to death? Does smoking lead to lung cancer? Does if you don't wear condom and you have sex with people, multiple sex, will you be able to get HIV? Does salt intake predispose somebody to hypertension? So that is causal relationship. We are trying to infer causation. So does this lead to this? Does this lead to this? That is causal relationship. So that one we infer causation. So with those kinds of studies, we look at the case control studies, retrospective studies, prospective studies. Prospective is the same as cohort studies. Retrospective is also the same as cohort studies. We will look at them. That's what we have the follow up. Then we randomize clinical studies. So with this descriptive study, they just describe what are the percentage of when you come to regional hospital, and we are looking at disease distributions, or we are looking at professions. How many laboratory people are in regional hospital? How many disease control personnel or staff are in regional hospital? How many this? Those ones are what? Descriptive epidemiology. They are just describing. But they are not they are not asking that who did this? How many anemia cases are there? How many hypertensives are there? How many? These ones are descriptive. But if you say what led to this, what caused this, what led to increase in anemia cases, what are the factors that led to this, that becomes what? Analytical studies. So with the analytical studies, you have the case control, cross-sectional, retrospective cohort studies, prospective cohort studies, randomized control trials. But some people, some of the time, they add the cross-sectional to descriptive studies. So I guess you understand. Okay, so let's go to more explanation on it. So we have okay, so for the epidemiological, there are all these study designs are under epidemiological studies or public health studies. So we have descriptive studies, and the descriptive we have the case report at, like I said, and case report looks at when a doctor is reporting, like the HIV, when they saw that abnormal finding in the person. So they were confused. The doctor was confused, so the doctor had to write a report on the signs and symptoms and the history and the medical or clinical presentation of the patient that the doctor was examining. So when he brought the report, the results, the report was, or the findings were a weird finding, very rare. It has never happened before. The person became immunocompromised. Some of the time you even give the person drug and the person will die. So you have to report it that this is an abnormal finding in medicine. So the doctor will write a report and publish in a journal. So case report is on only one person. But if the doctor is reporting on more than one person, then it becomes a case series. More than one person. So this one, we don't need any sample size calculation for this. But if the doctor, for descriptive surveys, we have ecological studies, which maybe we compare the rate of hypertension in Sumrungu compared to Upper East. So if you look at hypertension in Sumrungu, hypertension in Boga, hypertension in Zwarungu, hypertension in Boko, then we look at their percentages and compare them. So this way we are not comparing individuals, we are just comparing communities or nations or societies. That one becomes what? An ecological study. So when we say ecology is like somebody's ecological nature. It's like your community where you are, whatever. So you are comparing the communities but not the human beings. Then we also look at observational studies. Observational studies is a study which you go in and you observe what is happening without you changing anything in the study. So for instance, we have the cross-sectional study, case control study, cohort study. So with observational study, for instance, if you are working on when I was doing my second degree, I was working on anemia in pregnant women, but that one was a cross-sectional study. So I went in and picked samples for anemic pregnant women and non-anemic pregnant women, and I used a cutoff point of 10 to define anemia. So if your HB is more than 10, then you are not having anemia. But if you're a pregnant woman, your HB is less than 10, then you are anemic. So these were the groups. So I took 200 anemic and 200 non-anemic pregnant women, and it became a cross-sectional study. With this, I didn't go and inject the anemic pregnant women with any drug. I didn't impose any restrictions on them. I didn't go and give them food to eat so that I will see their blood levels. I didn't put in any intervention. So I was just observing them, and at different time points I'll be collecting data, their blood samples, and analyzing. So that one becomes a cross-sectional. So for the cross-sectional study, at first trimester, I just took their samples. So just those who were visiting the hospital at their first trimester, I just took their samples and look at the differences in some iron status markers at first trimester, and also at the first trimester between the anemic and the non-anemic pregnant women. And that one made it very clear. I didn't impose anything. I didn't inject anybody with any drug. I didn't give anybody iron supplement or cyanocobalamin or anything to increase their blood level. And I made sure that these women were not under any intervention. Doctors have not given them blood transfusion or anything, because that one would have ended up in biasing the findings of the study. So we will later go into the case control study cohort. All the studies will tackle them one after the other, but I'm just giving a short exposition on them. So we look at the analytical studies. Analytical meaning that you do some analysis. So for the analytical study, the observational study is also an analytical study. I don't go and do anything inside, but I took their samples, the cross-sectional case control cohort. You will take their blood samples, and look for some markers, biomarkers in their samples. We can look for liver enzymes, AST, ALT. We can also look for blood group, MCV level, platelet level. So it makes it an analytical studies. So it's an analytical, but I didn't impose any intervention on the pregnant woman. But at the experimental, you have to impose intervention, meaning that most of the experimental studies, you don't do them in human beings because they are unethical. When you are developing a new drug, it's an experimental study. So you have a herbal preparation, you've taken to Center for Scientific Research into Plant Medicine or CSIR or Food and Drugs Board. You want them to analyze it for you. Such drugs, they will use laboratory animals and they will. So there is what they do, something we call animal models. So you use animal models for such studies. So what happens is that if you have the animal models, you have to induce a condition in them. So for instance, if you are looking for working on diabetes, so there is something called streptozotocin. My first degree, that's what I did. Streptozotocin induce diabetes. So I use laboratory animals. Then we have the eloxan. So for diabetes studies, these are the two drugs they are poisons that you give to the laboratory animals. You give to the laboratory animals in order to create an experimental diabetes in them, so that you can now give them the drug that you have developed. And the drug that you have developed is what you are going to use to treat these animals after you have induced diabetes in them. So if you want to do something on liver, we have something we call hepatotoxicity studies. So there is a study we published, myself and my colleagues in the Department of Pharmacy, the School of Pharmacy in UDS. We published a paper in November of last year, and it's on hepatotoxicity. So we use CCL4 to induce. So we use CCL4, or it's also called carbon tetrachloride. So with the carbon tetrachloride, we used it to induce hepatotoxicity. So we use it to cause hepatitis, hepatotoxicity in laboratory animals. So for the laboratory animals too, if you want to do a study on them, you will need ethical approval. And the University of Ghana, Legon, they are the only institution in Ghana that can give you ethical approval for laboratory animals. Last time when we asked for ethical approval, it was $4,000 for laboratory animals. So the human oneself is very, very cheap. Now Bronco research, it will be around 450 or 600 Ghana cedi for masters, and the first degree will be 350 or 400. But for laboratory animals, it will be around 4,000 will be around 120 million or so. For you to use just 15 laboratory animals to do a study, you pay for ethical approval, because we don't want to kill the animals and extinct their population or kill their population. So as a matter of summary, for experimental studies, we use, if you are looking for diabetes, we use streptozotocin to induce diabetes, or eloxan. And so this drug will go and act on the islet of Langerhans, or the beta cells of the islet of Langerhans, that produce the insulin, and it will destroy it. Then you use the drug, the herbal drug that you have prepared, or the allopathic or orthodox drug that you have prepared, or that is in the market or from outside the country, to manage the condition in the laboratory animals, or to reverse the diabetes in the laboratory animals. Whereas if you are looking for liver problems, you use carbon tetrachloride to induce hepatotoxicity, and then afterwards you give your drug of choice and see whether the drug can be able to cure the hepatotoxicity. So the drug that you can use over here, most of the time the drug we use to cure hepatotoxicity is called, the drug for the hepatotoxicity, we call it, what do we call it? Selimarin. So you realize that people with hepatitis B, they use the Selimarin. That's what they use to prepare Born Leaf, Livormin, and the others, Hepantiva and the others. So let's proceed. Please, some people are joining. Okay. So let's proceed. Okay. Okay. So let's proceed. Okay, so types of this image will explain the study design better. So for for uh, so we have non-experimental or observational studies. So for the observational studies, we have the any experimental you see or interventional studies means that it's either a randomized control trial, and randomized control trial is also called a clinical trial. Then a non-randomized control trial is also a clinical trial. You are given an intervention, but over here you don't randomize. Randomization means that you the one doing the experiment, like you don't go systematically. Like you if you have 100 laboratory personnel at the regional hospital, and you come and you are sharing toffee, you just give the toffee to anybody without you saying that this is my family member, this is my sister, let me give her first. There is no favoritism. You have to even cover everybody's eyes. When you cover everybody's eyes, so if the investigator covers his eye and he doesn't know whoever is going to give it to, and the investigator covers his eyes, then the respondent, anytime you hear respondent, is the same as participants or study subjects. So when the respondents also, if their eyes are covered, we call it blinding. Blinding. So if we blind only the investigator, that is you who is doing the research, if we blind you, then we call this, and we don't blind the participants, or we blind the participant and you don't blind the investigator, we call it single blind. Randomized control single blind study. What is happening? So we call it single blind study. Then if we blind both investigator and so single blind study. That is when you blind either the investigator or the respondent. But if you blind both of them, you blind the investigator here, you blind the respondent at the same time, we call it double blind study. So in the end we have what? Randomized means that you are assigning whatever test or whatever your investigation you are doing. You are not being biased. You just come and you distribute at random. So we have the randomized single blind study. Then we also have the randomized double blind study. So double blind means that the investigator does not know the subjects he is dealing with. So if you want to do blinding, that means that you, the investigator, you shouldn't use the names of the subjects that you are recruiting. Then the respondents also, they don't know each other's findings, and they don't even know what the study results, whatever they are going to use it for, because their names are not there. So when you publish the findings, the respondent does not know that his information is even in the internet, because you didn't use his name. He doesn't know what is going on. The investigator also does not know the names of the respondents, so he doesn't know that if this one is for you or this one. So we have the randomized randomized single blind study and randomized double blind study, or we call it double blind control study or whatever. So these ones are the experimental or interventional study. So we can see, so let's go here. So when we come to the non-experimental, meaning that you are not going to administer a drug or anything, or going to administer a condition. So over here we look at the population-based study. An example is the descriptive health study, which the Ghana Health Service has been using often. We also have ecological study. An ecological study, like I explained initially, is a study that compares large groups of people instead of individuals for differences in things such as cancer rate. So if you want to compare the prevalence or the levels, percentages of cancer in Ghana, in the 16 regions of Ghana, look at the, or even HIV, percentage of HIV in the 16 regions of Ghana. We look at Upper East prevalence, Central region prevalence, Eastern region prevalence. So that becomes an ecological study. Then we go to individual based study, because these ones are population based. So we go to an individual based study. And the individual based study, we have the descriptive studies. That is, you are describing what is happening. So we have the case report, case series. That's what I said, that the doctor will find an abnormal condition like HIV in 1983. So the patient even died. Some are saying the patient is still alive, some are saying he is dead. But those times there were no drugs. So the doctor described it, whether the patient died or the patient was alive, the doctor will capture it in his report. That becomes a case report. So when you go to some universities, most of the lecturers, especially those who are medical doctors, they have been using case report for their promotions. They don't want to spend their money in doing research. So the case report that they publish in the journals, that's what they use to go for promotion, which is a bad thing, because we're looking at the case report here. That is the minimum study that carries weight. That's the list. From case report, you go to case series, then ecological study, cross-sectional study. So the randomized clinical control study is the highest of the study. If you do clinical trial, that means you are going to discover a drug. You are going to do, so they respect your findings, and that is what most of the granting agencies or the funding agencies, that's what they, most of the time, they finance those scientists, because you are going to really bring about, like you are discovering a COVID-19 drug. Drug discovery takes about 13 years to discover a drug. Paracetamol took about 13 years to discover it. And all this, they will use in vitro studies. That's inhibition studies, then that's test tube and other things, cell line, cell culture, and all those things before they come to animal models. Then they come to human, before they extrapolate their findings to human beings. And even the human beings, they will take about 10 people. If the drug does not kill them or give them problem, then they will increase the number to 100,000. So all this in about 13 years before they can say paracetamol is safe for people to take. So looking at, let me explain. I'm dwelling much over here because it will make my the lecture very, very simple. So for when you finish the individual, so when you finish the case report, the case series, then you go to the analytic. So the analytical study, most of us in Ghana here, most of the researchers in Ghana, apart from the pharmaceutical, those who do pharmaceutical research, they have been using the clinical trial because they are always working on drugs. But for we in the health sector, most of the study we do is the cross-sectional study, and cross-sectional study is also a prevalence study. So anytime you hear cross-sectional study, is prevalence. How many abortions or the prevalence of hypertension or diabetes in Upper East region in the year 2019, whatever. So cross-sectional study is the prevalence study. So it doesn't carry much weight. Then case control study, we pick cases and controls. So if you pick about 200 anemic, 200 non-anemic, and you look back to find out what conditions or what exposures cause them to have the anemia. then that one becomes what? Case control or case reference study. Then the cohort study, you pick people without a condition and you follow them up until they develop a condition. For instance, when you are looking at gestational diabetes, when you are looking at gestational diabetes, people with women with gestational diabetes, most of the time, they don't enter into pregnancy with the condition, but they develop it along the course of the pregnancy. They develop it along the course of the pregnancy. So, with such women, you cannot say that the study becomes what? A longitudinal. Longitudinal is the same as cohort. Longitudinal study is the same as a cohort study. So anytime you see longitudinal, is the same as a cohort study. So let's see. So if you have gestational diabetes, the woman entered without gestational diabetes. She didn't have gestational diabetes. Then, so not having gestational diabetes. Okay. Then, after 24 weeks of gestation, she develops gestational diabetes. So, when you enroll at enrollment, that's the first week when she enter. At the first week when she enrolls, maybe within week 7, week 2 to 5, when she enrolled in the study, she didn't have any diabetes. Gestational diabetes is a condition that you acquire when you are pregnant. That is if you have a predisposition factor or a genetic factor for it. So you have genetic predisposition for it. So anytime you are pregnant, you alone, your blood sugar level goes up. That is what we call gestational diabetes. So week 2 to 5 weeks, this woman did not have gestational diabetes. But from the 24th week, she developed the gestational diabetes. So when you are following such a woman, you make sure you pick her sample from between 2 to 5 weeks, because then she doesn't have the condition. Then afterwards, you pick a sample when she developed the condition at week 24. So now you can compare the samples you pick at week 2 to 5 weeks, and the samples you pick at week 2 to 5 weeks becomes the baseline samples or specimen. Let me make it specimen, because in statistical analysis and those things, if you bring sample, it becomes your participant. So baseline specimen. Then this one becomes what? The intervention, the specimen that you take when the condition developed. So this is the meaning of. So here, over here, you can choose to compare the baseline, those who did not have the condition with those who did not have the condition. Take the sample at baseline, then you also pair it with somebody. Okay, so baseline, then when the person developed the condition, you can pair it with a control, people who did not develop the condition. So over here, if it's at the 24 weeks, if you want to go into case control study, then you shouldn't take any baseline specimen. So you delete the baseline specimen. So if it is a case control study, then the woman developed gestational diabetes at week 24. So the 24th week, when you select them, you are going to get those who developed the condition and those who did not develop the condition. So those who develop the condition are going to be controls, the cases. cases. So if you have about 200 cases, 200 cases, then the controls will also be what? 200 controls. 200. So for gestational diabetes, the cases here are those who did not develop the gestational diabetes, and those who developed the gestational diabetes are the cases, and those who did not develop the gestational diabetes are the controls. So you follow the women, or you go to the hospital and say you want women who are aged 24 weeks, or you can still open it and say 20 to 24 weeks women, like their gestational age is between 20 to 24 weeks, and they are either having gestational diabetes or not. So those who are having the gestational diabetes at between 20 to 24 weeks, they become the cases, and those who are not having the gestational diabetes at week 20 to 24 weeks, they become the control groups. We can go back now and continue. So I've explained the case control or case reference group. I've explained the cohort study. So the cohort study over here, like I said, you take the specimen at baseline and follow them up to along the course of the gestation to find out when they develop the condition. So with every cohort study or longitudinal study, the subjects begin their study without the condition, then they end the study. So they start without the condition. So we take baseline samples when they don't have the condition. Then when they develop the condition, you make your diagnosis and take their specimen. Then you find the difference between the baseline results or analytical results and also the stage where they develop the condition. You take the specimen, analyze it, and find the difference or causal association between them. So we have explained the randomized control trials, that's the clinical trials and most of the time ethical approval of clinical trial takes a long time and is very, very expensive, and that's what led to the drug discovery and other things. So those are interventional or experimental studies. Okay, so let's zero in on the real lecture. So the study designs, like I've explained, we have observational study that the researcher studies. It's also called the researcher studies, or the researcher studies does the studies but does not alter or change anything within the study. So I explained to you that you don't need to give them drugs or anything, but for the experimenter you have to even give a drug that induces a condition. Then after using that chemical or the drug that induces the condition, then you now use your own drug that you have developed to cure that condition. That becomes an experimental studies. That becomes an experimental studies. Lister, please, I'm presenting, so join the class, please. Okay, so in observational studies. Okay, so I'm explaining. So this one I've explained already. You can go and read it. It says observational studies. The researcher observes and systematically collects information, but does not try to change the people or animals or reagents being observed. For example, a survey of drinking habits among students. So if you want to determine how students drink, or a researcher who joins a biker gang to study their lifestyle. So with this, you just observe, but you don't change anything in the sample or the population or the participants that you are doing the study on. You don't change anything. When I was doing the study on the anemic pregnant women, I didn't give them any drug. I didn't do anything to them. I just picked their samples and compare anemia and non-anemic. So note, as long as the researcher does not try to change their behavior, it is an observational study. It is an observational study. It is an observational study. But I've explained the experimental studies. That's when you apply, when you intervene in the study. You apply a law student, or you deceive a law student and give the law student a beer to see whether lawyers argue better when they are drunk. So you give a law student a beer and let him go and work on a law case to see whether he can argue better or not. You could have, if it's observational, you could have just, you wouldn't need to give the law student or the lawyer any beer, and just go and observe how he does his work. But this one you have to induce the person with beer. So now over here you are an ass. You are inducing somebody with money to see whether he's corrupt or not. So that one becomes an experimenter. You the experimenter, or you the investigator, you go inside and influence things. So encouraging bikers in one group to stop smoking cigars to see whether they get less belligerent. Warning one group of students that you are going to take blood alcohol levels. So if you tell the student that they are going to take alcohol levels next Monday to test for alcohol, and comparing their levels to another group that you did not warn. So if you warn them, if you warn them they are going to take blood alcohol level, none of the students will drink. They will not drink. But if you don't warn them, then you will now find out those who drank and those who did not drink. Then you can be able to justify your fight. But if you tell them, they will not drink, all of them will not drink, and they will come. So that is so warning the students becomes an experimental study. But if you don't warn them, it becomes observational. So when do you do an observational study? That is when you merely want to collect descriptive information. That is the evidence of diabetes rising? So you look at the prevalence. When you want to find out why students do not attend lectures, that's your reason, your hypothesis. When you can't do an experiment, because experiment is very expensive. The ethical approval alone for experimental study, like I told you, the laboratory animals, ethical approval alone for Lagos is around 120 million. So can you afford such a money? If you cannot afford, just go to observational study and observe your participants without giving them drug and other things, and do your own work. How fast does the earth move around the sun? So for instance, if astronomy want to find out how fast does the earth move around the sun, some of them, this way you can't do an experimental study. It's not possible. You can't go and experiment with how fast the earth moves around the sun. It's not possible. When it's not, another reason why we do an observational studies, when it's not acceptable to do an experiment. For instance, you can't do an experiment when or to find out how much not wearing a condom increase the likelihood of HIV infection. So you come and tell people in Bolga that I'm doing a research to find out if you don't wear a condom, will you get HIV or not? So you recruit 100 men, go and get prostitutes for them, and say that go and sleep with the prostitute and let me see whether you can have HIV or not. It is unacceptable. So you just need to do observational study. With the observational study, you don't influence who go for HIV person or not. Yours is that you come around to see those who are sleeping with prostitutes, men, married men who go about sleeping with slave queens and prostitutes. The rate of HIV in them compared to married men who don't go around sleeping with anything or chasing anything in skirts. You don't go around chasing women to sleep with them. You look at the HIV in those men, and those who go around cheating on their wives, you look at the prevalence of HIV in them, and this one becomes an observational study. But if you are telling them that they should not wear condom, go and sleep with women outside and see whether they will get HIV or not, it is an impossible study. You can't do an experiment with that. Okay. So let me pause and save this recording.